Good afternoon and welcome to the University of Manitoba Alumni and Friends Virtual Learning for Life Alumni Scholars Series. My name is Tracy Bowman. I'm the Director of Alumni Relations here at the University of Manitoba and also a proud alumna and the moderator for today's session. Thank you for joining us and making this part of your day, wherever you are joining us today. This series of the Virtual Learning for Life program is especially exciting because we have the opportunity to profile many of our University of Manitoba alumni who are doing amazing things and excelling in every sector imaginable all around the world, proving that alumni are one of the finest examples of how a degree from the University of Manitoba helps better our world. So before I get started and introduce today's speaker, just a few housekeeping details. You're all viewing this on YouTube as usual, and we're recording today's session. And it will be up later, a little bit later today or tomorrow, where you can view it as many times as you'd like again, as well as share it with your friends. Again, as usual, we use the platform Slido. That's www.slido.com. Password is VLFL24. And I encourage you to put that up as another window on your desk, on your laptop or iPad, so that way you can ask questions at any time. And please do ask those questions at any time our presenter is speaking, and I will facilitate those at the end, and we'll also post them up on the screen so all of us are able to see them. So now I'd like to introduce today's speaker, Taylor Morriseau. She's a Bachelor of Science honors grad from the class of 2017, and she'll be speaking on the topic of rewriting history, explaining the rise of type 2 diabetes among Indigenous youth. Now let me share a little bit about Taylor. She's a PhD student in pharmacology and therapeutics in the Rady Faculty of Health Sciences at the University of Manitoba, and is one of Canada's top graduate student researchers. She has received the very prestigious Vanier Scholarship in 2018, and she was also a Rhodes Scholar finalist, which is incredible. And in 2020, she was the recipient of the University of Manitoba's Distinguished Alumni Awards in the Outstanding Young Alumni category. And very recently, she's also the recipient of the Nellie McClung Foundation's Manitoba 150 Trailblazing Women. So a huge congratulations to you on that award, Taylor. It was just released a few weeks ago. So with that, over to you, Taylor.
Thank you so much, Tracy. That was incredibly sweet. And I mean, it's very humbling to be the recipient of the Distinguished Alumni Award and the Nellie McClung Foundation Award. So just thank you so much. And thank you to the University of Alumni team for really inviting me to speak today and to share my work with you all. And of course, welcome to everyone who's joining. I'm really excited to share this with you. So I've been fortunate to call U of M home for the last Bachelor of Science graduate from the Department of Microbiology, and now as a PhD candidate in pharmacology. And really, it's the experiences, the friends, the mentors, everyone that I've met at my time at the university through Migiziaga Mek, the Indigenous Circle of Empowerment, Ongamazin, Dream, Krim, everyone here have been so supportive and have led me to be here today. And I'm really, really just grateful, and I want to say thank you. I also want to acknowledge Treaty 1 land and the relationships that I formed on this territory, which are so integral to who I am and what I do. So I'm going to take off my researcher hat today, and I'm going to try and instead tell you a story. And this story, as I've said, we're trying to rewrite history because this is a very long story and at times it has been misguided. And I hope by the end of this talk, you'll have a new appreciation, and you'll see the ways in which we are trying to understand type 2 diabetes among Indigenous youth in this province. So I'm going to start here in 1999, and this is when the British Medical Journal featured this headline, Thrifty Gene Identified in Manitoban Indians. And so although the text is small, the article reads, a study conducted in a reservation in Northern Ontario has identified a genetic mutation that seems to have allowed Indians there to survive famines in the past, but to have triggered diabetes when food became plentiful. So this, as it's known, is the thrifty gene hypothesis. And the thrifty gene hypothesis is not new. In fact, it was proposed by the geneticist James Neal in 1962, and it's based on this evolutionary notion that a particular gene allowed hunter-gatherer populations to survive periods of feast and famine. but it's become incompatible with our kind of modern sedentary lifestyles. Now, despite James Neal himself actually rejecting this theory, the racialized reincarnation of this hypothesis has really persisted, and it's infiltrated a lot of genetic research, particularly among Indigenous Australians, Native Americans, as well as in Manitoba and Ontario, where First Nations have been kind of the subject of this thrifty gene hypothesis. And this last publication here is the focus of the 1999 article. So I want to take a step back and I want to tell you how I fit into this story. And so as I mentioned, I graduated with my Bachelor of Science in 2017 in the field of genetics, and yet I had never kind of heard of the thrifty gene hypothesis. So how do I position myself as Kathy Absalon and Cam Willett Wright in Research as Resistance, and how am I accountable to the people who are, at the center of this research? And so, I am mixed ancestry Cree and English. My paternal roots are in St. Peter's Indian Reserve or Peguis First Nation, and my maternal roots are kind of Europe prior to World War II. I'm the first in my family to pursue a PhD, and I think my mom is watching today, and she knows that for me, doing this research and being here in this PhD is really about finding space and creating space for the next generation as well, and opening doors for people and underrepresented voices that may not have been heard before. So I'm really thankful. This journey for me has been about, unlearning, reclaiming, and remembering. And I'm really thankful too to the Indigenous scholars who've already paved such a way in this field. So I'll highlight a few of them in the next slide, but a few of them too are pictured here as members of the Summer Internship for Indigenous Peoples in Genomics, otherwise known as the SING Consortium, and who have really added another perspective to this thrifty gene hypothesis. So, as I said, when I started my PhD in 2017, these were some of the really foundational articles that told a different story of the thrifty gene. Together, they really challenged the history and the validity of this hypothesis. And if you have the time, I recommend reading them in full, but I'll try and capture some of the main points here. So what does it mean when we say the geneticization, for example, of diabetes, and how does that relate to the 1999 headline? Now, geneticization is, by very simple definition, kind of prioritizing genetics in the study of health and biology. So if we think about the 1999 article, we know that 1, genetic terminology was used to define the problem. 2, we know that genetic expertise were enlisted to deal with the problem. And 3, we know that the knowledge that was kind of gleaned from these early studies changed the way that we viewed this otherwise incredibly complex condition. So where geneticization becomes especially problematic is that it changes the way that someone can view themselves within their families or their communities, and again, how inevitable that condition then becomes. And perhaps most alarmingly, this literature is very complicit in ignoring the social and historical drivers of these complex diseases, and it pathologizes the individual rather than the systems responsible. So a few of the narratives that continue to persist in racialized genetics include using race as an independent risk factor. And so this is like a biological entity, much like the presence or absence of an appendix, which would then therefore be a risk factor for a condition like appendicitis. These studies are usually founded purely on biological inquiry, and they really grow excitement over this idea of finding and unraveling mysteries of disease while skirting around kind of the uncomfortable topics of colonialism or racism. And again, owing to this objectively scientific lens, these genetic studies enjoy a certain level of immunity to really cultural, political, or social input because they have the goal of identifying that causative gene that can explain that deficit. And yet, kind of researchers have in the past, and they continue to do this because of this quote unquote potential for benefit. And so the decades we know spent searching for a causative gene have really yet to provide any sort of changes in clinical practice or policy for any Indigenous population in the world. So lastly, geneticization serves to really ostracize the disorder, and it places the responsibility of that genetic burden onto the individual themselves rather than, again, the systems and the historical inequities responsible. So with all of this in mind, let's go back to the genetic variant in question that I told you about earlier. And again, this was first reported in northeastern Manitoba and northwestern Ontario within Anishinaabe or Oji-Cree communities, circled here in Manitoba in blue. And so you might recognize this as the Island Lake region. The variant itself is identified in this hepatic nuclear factor 1 alpha, or as I call it, and for short, HNF1 alpha, where a single amino acid change at position 319 of this protein changed from a glycine or a G to the variant S or a serine. And so even though it's a single amino acid change, if we want to look at the protein sequences from various different species, and you can see the mouse here or the rat here, this is position 319 of the HNF1 alpha protein. You can see a G is very conserved throughout all types of species. And so when we know an amino acid is highly conserved across species, this suggests a change in the protein sequence could be detrimental to protein function. But again, we actually don't know this yet. So what's important to note here too is that this genetic variant has only ever been identified in the Island Lake region of Manitoba and no other Indigenous or non-Indigenous groups that we know of. And so in this initial study, they also identified individuals who were unaffected, so who carried 2 copies of the G, or they compared these to heterozygous individuals who carried 1 copy of the variant S allele, and lastly, people who carry 2 copies of the variant S allele. Because if you remember kind of genetics, you inherit 1 copy from your mom and 1 copy from your dad. And so here they looked at the percentage of people who develop diabetes versus their age of onset. So you can see here that individuals who are homozygous, so GG, they typically, 50% of people with the GG genotype develop diabetes in their kind of mid-50s. Comparatively, people who are heterozygous for this variant, it seemed to accelerate the disease onset, and so they typically develop diabetes by about age 30, and people who are homozygous for the variant typically develop diabetes around the age of 20 and even earlier nowadays. So from here, let's kind of think about the clinical side of this story. And so this is in the early 2000s when clinicians really started to understand how this variant may impact diabetes and the clinical phenotype at the time of diagnosis. So in Manitoban youth, children who were homozygous, again for this variant, presented at diagnosis with higher hemoglobin A1C levels, which is a sign of higher blood sugar, and it's a common feature in diabetes. But interestingly, these children, again, actually had reduced BMIs at diagnosis compared to kind of the GG genotype. And so this is about 50 pounds less than their GG counterparts, and they also had reduced fasting plasma insulin. So what these results and what the clinicians kind of concluded from this is that weight, which is normally a factor associated with diabetes diagnoses, rather this lower insulin level suggested that there was potentially a problem in the pancreas and possibly a dysfunction in the pancreatic beta cells, which are found in the islet and which are the only cells in your body to produce the hormone insulin. So at the level of the beta cell, the question then became, what is HNF1 alpha doing at the cellular level? So the normal function of HNF1 alpha is a transcription factor. And as you can see here, HNF1 alpha in a cell looks like kind of like this, and it can be responsive to external cues. It can then bind to the DNA, and it can help to transcribe some of these genes, a whole bunch of different genes, but including those that are involved in things like beta cell metabolism, so mitochondrial function and the creation of energy, as well as maintaining kind of a normal beta cell that can secrete insulin and respond to glucose. Importantly, HNF1 alpha is indeed expressed in the pancreas, as well as other metabolic tissues. And I just want to stop here, though, and you might be wondering, like, where do we keep going? I mean, we've gone from the discovery to the clinical picture, and now we're really diving down into this cellular level. And you might be thinking to yourself, isn't she doing exactly what she just said not to do and not to hyper-focus on genetics? So I want to take a step back and say, how are we going to not contribute to the geneticization of diabetes, even though these questions still remain? So how do we change the narrative, and how do we effectively rewrite the history of this quote unquote thrifty gene? So first, let's open up the story to the larger socio-political and historical drivers of health inequality. And let's look at these variables that I haven't really mentioned yet and are currently missing from this research framework. To do so actually requires investing in research tools to really incorporate these complexities into the experimental design, and that better reflect the true history of Indigenous peoples in this country, in this province. So if we take a look at the TRC call to action, especially call to action number 18, this actually tells us exactly where we need to start on this process. Number 18 states that Indigenous health, including diabetes, is a direct result of previous government policies, including residential schools. And so by applying this historical lens, we should theoretically be able to pinpoint the precise colonial policies that are kind of responsible for present day realities. So to understand diabetes in Indigenous communities, we have to go back, and we have to go before 1999 all the way, let's go to the mid-1600s to start. So keep in mind that genetics is very slow to change, and the variant was likely present at some point in this timeline. And it's preceded the rise in diabetes, which is really important. So around the 1600s, we know that our ancestors lived on the prairies and used a really diverse set of food procurement strategies that included things like hunting, harvesting, trapping, as well as preserving foods that was in step with the seasonal and animal migration patterns. However, by the early kind of 1800s, we know that the expansion of the European fur trade by the Northwest Company as well as HBC placed a really high strain on wild food sources, particularly the larger animals, thereby increasing the kind of reliance on smaller animals like rabbits and fish. And so without these big game animals, the larger families and the larger social constructs really had to break off into smaller extended family units and survive primarily on things like rabbits and fish, rather than caribou and moose that were largely exterminated as far as up to the 1820s. So the Indian Act of 1876, which is still actually in effect today, also further restricted the use and the management of Indigenous lands. And for example, people who wanted to leave the reserve to fish and to hunt really actually had to request permission from an Indian agent at one time. And so you can see how this would completely undermine Indigenous food security and Indigenous food systems. So this disruption of Indigenous food systems caused some Indigenous leaders to have to cede their land for in exchange for treaty reparations and treaty provisions that were promised at the time to alleviate hunger. And we know some of these actually didn't come to fruition either. These communities were also hit extremely hard by the Great Depression, where we know incomes from the fur trade plummeted by as much as 66%. And it wasn't just hunger being driven by the absence of things like moose and caribou, as well as reduced income. We actually know the Federal Department of Indian Affairs cut unemployment relief by up to time. This is also the era of residential schools, and so we know systematic removal of up to 150,000 children. It happened between the years 1883 up until 1996 even. And the ultimate goal of these schools was to assimilate children into the Western world and sever the ties between families. And we know in these schools, children experience chronic undernutrition that was characterized by really insufficient caloric intake, minimal protein and fat, limited access to fresh produce, and this could be over a period of up to 5 to 10 or even more years. And so back in the communities themselves, permanent stores were established around the 1940s, again, changing the subsistence practices of the communities. And so although hunting and fishing and trading and living off the land are still very important parts of Indigenous life, this is the context in which modern communities were formed and how we know them today. So with these kind of rapid transitions that, from contact and onwards, the first diagnoses actually of type 2 diabetes were reported in the 1980s, again in Winnipeg. And it's not until 2015 that we kind of had a little bit of accountability. The TRC calls to action were released, and we're still not seeing any real changes. We have an increased awareness and a commitment from some government and some academic institutions to reduce the rates of chronic disease. But the rates of type 2 diabetes are actually still increasing in youth, and it's giving rise to these gaps that we have today. And so this was a stat that was released this year in which rates of type 2 diabetes in First Nations youth are 25 times higher than all other Manitoban youth. So I want to dig a bit deeper into this residential school era that I mentioned. And the work here of Ian Mosby and Tracy Galloway is really instrumental to understanding how patterns of chronic disease are a function of residential school survivorship. And so one of the things that not a lot of Canadians are aware of is that because malnutrition was so common at these schools, between 1942 and 1952, federal scientists and doctors actually conducted a series of nutrition experiments um, on children who were attending these schools. And this included things like vitamin fortification. So I encourage you to seek out this manuscript and read it if you have time as well. But even worse, the malnutrition that was caused by the residential school attendance, the TRC concluded that the federal government actually knowingly underfunded these kitchens and provided only half of that was required for a really nutritionally adequate diet. So collectively, these schools created long-term health effects for the survivors themselves. This included mental, spiritual, emotional, and physical consequences that arise from widespread abuse, and that included childhood malnutrition. From a cultural standpoint, the residential school systems alienated children from traditional dietary practices, and this effectively severed the transmission of knowledge to future generations, as well as reinforced patterns of nutritionally inadequate diets for survivors and their families. And so, kind of based on this work, you can see how colonization effectively stripped communities from their traditional food practices in a very short period of time. The transition from off-the-land foods, including moose and caribou and beaver and muskrat and fish, as well as wild plants and wild berries, towards more Western store-bought foods has dramatically changed the macronutrient composition of historical versus contemporary diets. And so you can see that here that carbohydrate intake in a historical diet could actually be as low as 5%, and that would be obtained only seasonally from wild plants and berries. Instead, fats were actually a particularly valued energy source because they're so nutrient dense. And of course, if you're living off the land, that would be incredibly valuable. So the transition to more processed foods, again, high in a different fat, so high in trans fats, high in refined sugars, as well as salt, at a price that's actually comparable, as you can see at the bottom here. They're kind of the same availability and the same cost as healthy foods, but these types of foods have really been, and this dietary transition has really been attributed to the rise in type 2 diabetes incidence as well. So in support of this, as part of the Sandy Lake Diabetes Health and Prevention, Sandy Lake Health and Diabetes Project, 500 participants who live in Sandy Lake and who did not have diabetes at baseline were then followed for 10 years. At follow-up, 86 of those individuals had developed diabetes, and trying to understand how food might be have identified 3 different dietary patterns based on a food frequency questionnaire. And so from this, they identified, they identified 3 different patterns, the first being balanced market foods, beef and processed foods, as well as traditional foods. And it was the beef and processed foods that were associated with the increased risk of type 2 diabetes, whereas balanced market foods and even less traditional foods were not. So again, understanding how these types of factors play into the diabetes picture and this larger picture of colonization, I want to present to you what essentially is the framework of my PhD. And so we've shifted the question from kind of this genetic problem to one that's informed by the history and the current realities that we've just covered. So my hypothesis centers on this interaction between traditional foods as well as genetics. And by designing kind of experiments around strength-based questions, we've shifted the focus away from the pathophysiology and the geneticization to understanding how genetics might actually be a positive here and how they might interact with traditional foods and kind of provide resiliency to living off the land. And so by modifying experimental diets, we can understand then how this nutrition transition has really impacted diabetes at a level that's way beyond the thrifty gene. And by situating this within the context of colonization, the results we hope would be far more amenable to things like community interventions as well as advocacy initiatives. But how do we incorporate this into like an actual experiment? How do we study genetics, diet, beta cells? How do we bring that all together? So, as I mentioned, I'm a basic scientist, and we've utilized kind of the tools that are available to us in order to bring this problem into the lab. And we've introduced the G31NS variant into two different model systems. And so the first are pancreatic beta cells. So these are beta cells kind of in a dish. These grow and they respond to glucose just like a beta cell in a pancreas would. And then we've also introduced this variant into the G319S expressing mice. And so this is a mouse model that we've used CRISPR-Cas9 on. And so you might kind of recall CRISPR-Cas9. It was in the news recently because the two women who pioneered this technology actually won the Nobel Prize for Chemistry last year. And so CRISPR-Cas gives us an ability to target a single base pair. And as you can see here, we've targeted this glycine and changed it to an alanine in the DNA. And this actually gives rise to this G319S variant at the protein level. So in order to kind of study these gene-diet interactions, we can also modify the experimental diet of our rodents and decide on what they get to eat. And so we modeled this after both traditional food nutrient intakes as well as kind of what it looks like today. And this involves changing the overall macronutrient composition, as I've kind of broken down here, but also the source of the nutrients themselves. So we've changed, for example, the fatty acid profile by incorporating fats from butter, for example, in the present day, compared to fats from things like fish oil or different animal sources. And so using this very controlled model system, we can really look closely at the level of the beta cell, at the level of the whole body, and things like insulin secretion, and understand how all of these things work together. So I want to share a couple of those results with you in the kind of the time we have remaining today. Just to note that these are all still unpublished results and the research is really ongoing. Our findings to date right now are in support of this idea that genetics and diet can interact either positively or negatively, and that affects overall physiology. So in this first experiment I'll describe to you, we've asked how this variant impacts, for example, insulin secretion. And we've isolated the islets from male mice, and we've done this through something called collagenase digestion. And we've performed a glucose-stimulated insulin secretion assay. So this means that we basically place these islets into solutions that either contain low glucose or high glucose, and then we look at how much insulin is actually produced. And so this kind of mimics what happens when we go from a fasting state to what happens when kind of after we eat a meal. And then looking at the results of the ELISA, we can then tell you how much insulin has been secreted by those islets. So looking at this graph, we can notice two different things. And so first, the amount of insulin secreted under a high glucose, or so that kind of glucose-stimulated insulin secretion response, is actually very comparable between all three different genotypes. And so this suggests impairing insulin secretion capacity. And impairments in this response are actually associated with diabetes progression. And so the absence of an impairment here is actually a very positive sign. Secondly, mice expressing the variant, so shown here in red or in yellow, actually tend to secrete less insulin in the basal or this low glucose state. And this could actually have really important physiological consequences that we're still trying to uncover. But one of the things we've looked at, and to understand how this might impact whole body physiology, we performed a glucose tolerance test. And so here we fasted the mice overnight, and then we performed this tolerance test in the morning, in which we inject a glucose bolus at time 0, and then we follow the blood glucose measurements over a period of 2 hours. And now the shape of the curve can actually tell us how efficiently glucose is cleared from the blood. And so you can see male homozygous mice in yellow, so kind of right here, actually tend to have higher blood glucose measurements over this 2-hour period. And so this would be kind of a glucose intolerant phenotype. Now, in comparison to the controls in black, our male heterozygous mice in red, so those that carry one copy of this variant, actually track nicely along with the controls. And so they actually do not show any signs of glucose intolerance here. So how does, the low insulin that I just mentioned, how does that fit into this whole body phenotype? Interestingly, when we looked at the amount of insulin secreted in parallel with this glucose tolerance test, as well as kind of right after fasting, what we can see in these male heterozygous mice in red is that there's lower circulating insulin throughout this entire test, as well as particularly at fasting. And so what is a lower fasting insulin? What is it doing to improve metabolic health, and how is it preventing glucose intolerance? We don't know how this all fits together quite yet, but one of the kind of lines of evidence we're following is that these mice have higher blood ketones. And so ketones are produced by the liver in times of low insulin, and they actually may be normalizing metabolic health. And so one of the things we think is happening is that by lowering insulin and suppressing this insulin in the fasting state, we might be driving ketone production by the liver, which could serve as a metabolic fuel that's obviously non-glucose metabolic fuel that potentially could normalize metabolic outcomes for, in this case, heterozygous, but not homozygous mice. So this is an area where we're still trying to kind of put all the puzzle pieces together, and more answers are to come in this. But other than ketone bodies, we also are really interested in understanding how fats are involved. And you'll recall that fats are really abundant in traditional foods. So here again, we performed a glucose-simulated insulin secretion assay, but this time we used our kind of pancreatic beta cells in a dish. And so this time we also changed the treatment. And so we subjected these cells to a 24-hour exposure to a very high saturated fat. So this is normally very toxic to cells, and unsurprisingly, when we did a glucose-stimulated insulin secretion assay, as I'll show you here, in the control cells in black, the insulin secretion capacity under high glucose was very impaired. And so this is what we would expect. Typically, beta cells do not like to tolerate a lot of excessive fats. But what you'll notice here is that cells who carried this G311S variant, so again in red, these showed really robust insulin secretion after the same treatment, suggesting that these cells are somehow resisting this high fat content. So when we tried to understand why, and we looked at cellular fuel metabolism, we did so by measuring respiration. We found that these cells, again in red, that carry one copy of this variant, tend to utilize the fats, and they tend to metabolize these fats either at the basal rate or when we force them to respire at their maximal rate. And so together, these results to us suggest that the variant is indeed protecting the beta cells against any sort of fat-induced stress. So provided this propensity for fatty acid metabolism, my current research is really examining whether this variant shifts metabolism to preferentially utilize nutrients that are abundant in traditional foods. And if by consuming these foods, can we prevent or delay any of these diabetes outcomes? So we're studying this by placing mice on a high-fat, low-carbohydrate diet, again reflecting the macronutrient composition of a typical traditional type diet. And we assess different outcomes that can tell us about metabolic health. And so this is the research that I'm currently working on. I'm hopeful that I can share some of these results with you soon as I, So of course, I'm really excited by this research. The opportunity to use model systems to understand gene-diet interactions in a way that's actually meaningful and inclusive of the histories that I've covered today. So importantly, and as critical scholarship has asserted, we do not aim to prescribe type 2 diabetes to a genetic deficiency. Rather, it's about opening up the research and asking questions in a different way. By integrating Indigenous knowledge on traditional foods in combination with some of these, basic science tools, we hope the outcomes of this research might inform nutritional interventions for children with diabetes, while ultimately supporting, community-led efforts to access these traditional foods. But as we look forward into the future, we know that improving Indigenous health, food security, and sovereignty means taking this research out of the lab. And so strengthening community capacity and strengthening community engagement, prioritizing community knowledge is really important. Traditional knowledge, community capacity, and cultural continuity are really integral to moving anything forward. Secondly, training the next generation of Indigenous researchers, clinicians, healthcare providers, and so on. This is key to aligning the research and health priorities with community values, translating the needs of different stakeholders, as well as ensuring that long-term culturally sustainable programs can actually happen. And so together, Indigenous-led and community-centered projects are really going to be what's going to move the dial, improve health outcomes in a way that genetics alone is simply unable to do. So in summary, I just want to leave you today with an appreciation that health patterns in Indigenous communities are a direct consequence of intergenerational and ongoing colonialism. Secondly, that fundamental research is just one tool that's at our disposal in which we can provide insight into kind of the fundamentals of health and illness. Third, that all types of research, even the research that I'm doing in the lab, must acknowledge and incorporate the larger sociocultural and historical drivers of health and equity, and bring them into the research framework when and if at all possible. And lastly, to understand that future interventions must be guided by priorities and Indigenous knowledge if and only if desired by the community. So I'll leave you with that. But if you are interested in this story and learning more about diabetes in Indigenous youth, I'd highly recommend reading this story, this novel, Diagnosing the Legacy by Larry Krotz. It chronicles, the research and the families living with diabetes from the first diagnoses in the 1980s all the way through today. And it's really a nice read. And finally, I just want to express my gratitude for the mentors, the friends, the colleagues, everyone in my life who have made this journey so special. So a big thank you to my supervisors, Dr. Christine Doucette, pictured here, and Dr. Vern Dolinsky, pictured here, as well as everybody who, is in the lab and who makes my days in the lab so exciting. Really, we are a lab family, so I just want to say thank you to those people. It's been a tremendous privilege to also learn from Indigenous scholars, from elders, from community members throughout this PhD. And I just want to note that I'm going to carry some of those experiences with me throughout, my PhD and beyond. And since this is kind of an alumni talk, I also wanted to shout out a few of my undergraduate mentors that are responsible for sparking this love and passion for research. And so the first is Dr. Ayush Kumar, pictured here. We did this photo shoot in 2015, maybe. And I don't know why we're looking at this pink tube of pink rack of tubes, but we are, and it was featured quite a lot. So I just thank you so much to Ayush Kumar, to Ben Albenzi and Dr. Paul Fernyhow, who were my first co-op supervisors in the Department of Microbiology, as well as to all of the faculty in the Fuller Biological Building, especially, again, to Dr. Deb Court, who was the person who put me forward for the Distinguished Alumni Award and who's been such a supportive figure in my life since then. So with that, I'd just like to say, obviously, thank you, Ekose. I would love to take any questions if you have, either now or, of course, I put my email or my Twitter handle on the screen if you want to get in touch with me later. So thank you.
Thank you so much for that, Taylor. And so I encourage people, please contact Taylor afterwards, as she's put her email out there. So that's wonderful. But as we're waiting for questions to come through to Slido, so just a reminder, it's slido.com. The password is VLFL24 for the date. So I just wanted to, I'm going to start off some of the questions, if that's okay, Taylor, while we're some questions are coming through. And what struck me with, everything struck me with the research. It was quite interesting. I'm glad you're doing this research, as it's really important and an area that we need to know more about, and educating the community as well. Both non-Indigenous and Indigenous communities is really important. But when you were talking at the end in terms of some of the, the so what's, sort of what's next, that kind of thing. And you were talking about, I took a few notes, you're talking about, words like building capacity and training the next generation of Indigenous research researchers, and including traditional knowledge. I'm wondering if you can share a little bit more about some of those examples about what you mean by, well, like, what does building capacity really mean? And what does, using forms of traditional knowledge in terms of through your research and sort of what you see as next steps, or like the researchers of tomorrow, as you're saying, what would you like to see? Or if you could just maybe explain that a bit more.
Definitely. And I think that's a really exciting area. And community-based participatory research is, I think, the future of this type of knowledge, and incorporating Indigenous perspectives and incorporating Indigenous leadership. Indigenous-led projects should be the way that we're going to approach Indigenous health problems right now. And so building community capacity is not simply just bringing more research into the community, but it's asking the right questions, the ones that are going to make the most difference, and bring the impact where it's actually needed. And so that's where community-based research is really, really important. And then I say training the next generation, because we need people who can understand these different areas. And so Indigenous genetics is such a growing field. And I mentioned the summer internship for Indigenous Peoples and Genomics Consortium. This is one organization that's working, for example, to bring Indigenous youth into the field of genetics, and to tell them and to teach them both the pros and the cons, and how this could potentially be utilized to make a difference in our community, and so that we're not being kind of left on the sidelines. So that's just one area that capacity needs to be built in. But I think from healthcare providers, from clinicians working in the community, from people doing the research and asking the right questions that are based on the community priorities, this is how we're really, like I said, going to move the dial on diabetes. And I think the focus for too long has been on like a single gene, for example. So it's really about opening it up to different perspectives, and then finding things that are going to be the most impactful for the communities themselves. So.
Okay, wonderful. Thank you. So we have some questions that will start to appear on your screen. So the first one is, during your research, have you come across any information that suggests lack of physical activity or sport that affects the diabetes rates in Indigenous youth?
Yeah, so physical activity is obviously associated with diabetes, and or lack of physical. So sedentary lifestyles are associated with diabetes onset. We don't look at physical activity directly, I shouldn't, but it is obviously a possibility that changes in lifestyle, as I've gone through the entire history, we know that obviously living off the land and hunting and fishing, and these are all very energetically demanding. And so from this nutrition transition also happened kind of a transition in how much movement and daily movement that people are getting. And so it's kind of hand in hand with the carbohydrate content, and kind of the nitty gritty that I'm looking at as far as diet, but that is definitely another consideration that will need to be brought into this research, again opening it up to different perspectives. One thing that we are really interested in looking at too is how fasting, for example, is actually a beneficial aspect of particularly in the context of this variant, how fasting might actually be beneficial to metabolic health, because of course we know fasting is very common, both culturally. It's become very common in things like intermittent fasting and different kind of lifestyle changes today, but how this might actually interact with the variant as well, and in a positive kind of
I think Taylor's frozen just for a moment there. I'll ask the next question, and that is, thank you, great talk. Do the mice containing the variant have normal or even elevated alpha cell glycogen secretions? I don't know if you're able to hear me, Taylor. We had a few little freezing moments within the presentation today. Chris or Luke, are you able to help Taylor in any way? I'm not sure whose end this is. Okay, maybe we'll bring Taylor back in a moment. Encourage you to keep the questions going. If she'll be back in just a moment. She'll reconnect. I think she had some trouble there with her internet connection. But if you want to continue adding some more questions within Slido, we can certainly ask them of her, both during this presentation as well as afterwards if she's not able to connect. So we'll just maybe wait another 10 to 15 seconds to see if she's able to join again. And just keeping track of some of the support from our IST to see if she's able to connect. Oh, another question has come through. Thank you, everybody. Glycogen. Yes. Oops. See if there's any other questions that have come through. Well, I'm just getting some feedback through our technicians, and she still hasn't been able to reconnect. So again, I'll wait maybe another minute or so. I think she's still trying to connect. But as we're waiting, I'll just mention about next week's presentation, just in case you haven't signed up for that. And I want to add a little bit of context to it as well, because the topic title is very vague, and I think that's done for reasons. And so it's our alum John Amadick, and he lives in the United Kingdom, so it'll be a few hours ahead of time for him in the evening when he's presenting. But his topic is on out of the dark and unusual projects, and what that means is John is an engineering grad of the Price Faculty of Engineering here at the University of Manitoba. He's a retired oil, gas, and energy professional, and he was an engineer behind the dramatic rescue of nearly, if you remember, 10 years ago or over 10 years ago, of nearly 36 trapped miners in Chile. And that's just one of the many interesting projects that he has dealt with over his very distinguished career. So he'll be sharing about this particular rescue mission and others all over the world that he has experienced. So I encourage you, if you haven't registered, please do that. On our website, and as I said, this session will be posted in a moment. So Chris and Luke, I assume that she hasn't been able to reconnect. No, nothing yet. So there's a few more questions that have come through. I'll make sure to send them to Taylor, and we'll see if either she's able to write them out or maybe she's able to post a little video. We'll add that to it. And so there's also possible that there could have been a power outage. I'm very sorry that you weren't able to ask all of your questions. I hope you agree that her presentation was fascinating. She's doing some really important and impactful research here at the University of Manitoba, just demonstrating, as I mentioned earlier, the wonderful alumni that we have and graduate students at the University of Manitoba. So with that, thank you very much to everybody for joining us, and we will see you next week.